welcome once again to the Green Era Hub webinar series. My name is Barry Crosby, and I'm one of the communication officers for the Green Era Hub project. If you've been with us here before, you know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme, and it currently represents 15 EU ERA nets, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here in October 2023, and we've had 5 very successful series so far, focusing on circularity in agriculture, biotechnology to advance sustainable solutions for bioeconomy, sustainable food systems, precision agriculture technologies for a sustainable future, and plant health and protection. If you're interested in listening to the talks related to any of the topics from these series just mentioned, please do note that our recordings are now available to view on the Green Era Hub YouTube channel. So today we're going to continue with Series 6 after a short break for the summer holidays of our Breakfast Club webinar series, focusing on the topic of animal health and welfare, which, if you've joined us before the summer, features expert-led discussions aimed at fostering a deeper understanding of sustainable strategies for improving animal health, preventing disease, managing pests, and strengthening the connection between animal, human, and environmental well-being. And just a few housekeeping announcements. Please do feel free to type your questions in the Q&A chat section, which I've enabled for everyone. I'm going to do my very best to address as many questions as possible within the time that we have this morning, which is 1 hour. If at the end of the session you have a question for either of our speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Dr. Catherine Wirtz and Dr. Mariette Ducotet, and we'll open our session this morning with Catherine Wirtz, a senior microbiologist and immunologist specializing in host pathogen interactions and bacterial immune evasion at the Institut Pasteur in France. As head of the innate immunity and Leptospira groups, Dr. Wirtz work focuses on investigating how pathogenic Leptospira, which is the bacteria responsible for leptospirosis, interact with and circumvent the innate immune system. At the Pasteur Institute, her team develops innovative infection models, including acute and chronic murine systems and bioluminescence bacteria for in vivo tracking to study disease progression and test new host-directed therapies. Dr. Wirtz has published nearly 100 peer-reviewed articles and is internationally recognized for her contributions to the study of immune evasion by bacterial pathogens. She also plays a very active role in mentoring early career scientists and collaborating on infectious disease research across the Pasteur network. Today, Dr. Wirtz is going to be speaking about her research project entitled Comparative Host and Species-Specific Immune Responses to Macrophages Infected with Zoonotic Leptospira interrogans, which is part of the EU-funded ICRAD initiative. That's the International Coordination of Research on Infectious Animal Diseases. Catherine, thank you very much for accepting our invitation to our webinar series. I should just also point out that Catherine is joined by her colleague, Dr. Megan Fernandez, also from the Institut Pasteur in France. So we're extremely delighted to welcome both speakers and very grateful, in fact, that they can join us today. So I think without further ado, I'm going to stop sharing my screen now. And Catherine, Megan, you can start sharing your presentation. Thank you very much.
Hello, everyone. Thank you very much, Barry, for this nice and kind introduction. So I am coordinator of this ICRAD project entitled Leptimmunost, about Leptospira interrogans, so pathogens you introduced responsible for zoonosis. So just to tell you about leptospirosis, so it's a spirochete bacteria responsible for a zoonotic disease affecting all vertebrates and mammals, and it's a worldwide reimaging disease. The bacteria are mobile and can infect all kinds of different hosts through skin or mucosa. The cycle of the contamination occurs through rodents, rats, or other animals that are colonized in their kidneys. They are excreting the bacteria in the environment and contaminate other animals. Some of them, what's important that every animals can be infected, but manifests different clinical syndrome or symptoms. So in particular, like bovine and ovine can be infected and have genital tract infections, causing abortions. In horses, it's peculiar. It's more uveitis, and humans are very susceptible to the disease. Or they can have asymptomatic disease, but it can also lead to a severe and called the Weil's disease with multi-organ failure, and it can also lead to death. So my question was why this bacteria can lead to different susceptibility and symptoms according to the different hosts. And my approach over the years have been to study the cell wall of this peculiar spirochete. So they are of the same subfamily as Borrelia or Treponema, but they are really different in the fact that they have a layer of lipopolysaccharide. This layer of polysaccharide is responsible for more than 300 serovars, which really complicates the vaccine development. So they have this layer of lipopolysaccharide. They are characterized as the other spirochetes by a very, very abundant amount of lipoproteins in their cell wall. And because they are mobile, they are also, like the other spirochetes, they have an endoflagella, which is in the periplasm and very particularly is not protruding outside of the bacteria. So we are interested of all these components, so lipopolysaccharide, lipoproteins, flagellins, and other. And those are defining what's called the MAMS, microbial-associated molecular patterns, that are recognized by the host through macrophages or leukocytes expressing different pattern recognition receptors, such as toll-like receptors, which are the interest of today's talk of Megan, but also internal receptors or lectin receptors. So I have been interested over the years with this interaction, and we have shown different host susceptibility and peculiar interaction characterized in human and mice. So in this project, we hypothesized that altogether, the leptospiral recognition or escape from these pattern recognition receptors shapes the inflammatory response and would drive the host-specific pathophysiological consequences of leptospirosis. So we want to compare the host and species-specific immune response of cells infected with zoonotic Leptospira interrogans. And our specific aims are to study the interaction of TOL2, TOL4, and TOL5, and also lectins from several animal hosts with different serovars of Leptospira interrogans. So for this, we have a very nice consortium. So we have on board an Argentinian team led by Karina Caimi, and she's specialist of bovine leptospirosis and genomics. We have also, we are close collaboration with Dirk Verling's team, who is specialist on evolution of toll-like receptors and is a vet, really interesting also in bovine disease. We have also a Lithuanian partner, Česlovas Venclovas, who is a specialist of computational structural biology and is designing new software programs to study the interaction between these MAMs and PRRs. And finally, we are associated partner with a Canadian team and Elena Petrosova, who is specialist of lipid A. So lipid A is interacts with TOL4, and she's specialist of this structure, which are very difficult to study. So in this program, we hired two postdocs, so Nathalie Verling and Megan Fernandez. And we have several work packages. But for the sake of time today, I will not speak about macrophage responses, nor about the lectin projects, but we will be presenting, and Megan will present the work done on the interaction between LPS and TOL4, TOL2 and the bacteria, and also the flagellin. So what we hope as a whole in this project is to bring novel data on the relationship of Leptospira with their host. It has been studied only on an immunological side with mice and human. We don't know almost anything about the other host. We want to shed light on the diverse symptoms of leptospirosis observed in various animals. So it's an ambitious goal, but we think we will have some hints about that. And we want to set the basis of new perspectives of host-directed strategies to stimulate the innate immune system and overcome the different leptospiral escape mechanism by using these toll-like receptors agonist. So now I will leave the floor to Megan, who is postdoc in our lab and in close collaboration with Dirk's lab. Thank you very much for attention.
Oops. Yeah, so hello everyone. So as Catherine said, I will present you my work in the lab. So I work on the analysis of the recognition of different Leptospira strain by the TLR2, TLR4, and the TLR5 from various animal species. And as Catherine said, it's the work of me, of course, and Nathalie Verning close together. So just to show you some introduction about the leptospirosis. So in the lab, we work with 4 different strains. So 3 of them are pathogens. So we work with Leptospira interrogans that I call fiocruz, Leptospira interrogans manile, and Leptospira interrogans verdun. All these 3 strains are coming from the human clinical sample. But we work also with another strain, the Leptospira biflexa pathoc, that come from the fresh stream water. So it's the strain that we can have in the environment. So as Catherine said, in the cell wall of the bacteria, we can find some lipoprotein recognized by the TLR2, the LPS recognized by the TLR4, and the endode flagella recognized by the TLR5. And what was the purpose here? It was to obtain all these receptors of all the animals that we want to study. So here in the livestock, we want to obtain the bovine, the equine, and the porcine receptor. For the rodent, we want to obtain the mice and the rats and the hamster. And of course, we want to study also the receptor of the human. So to do that and to obtain the TLR expressing vector, we start with the tissue or blood of animals, and we need to extract the RNA to do the cDNA. After that, we of course have a lot of steps to go to the final product, but to just sum up, it's when you obtain your product, so your TLR sequence, you need to ligate it in the cloning vector, select the clone that express your good sequence, and after that you can ligate the sequence and your TLR in the PUNO1 expressing vector, and with the transformation and the purification you can obtain the final product, so the PUNO1 TLR depend of the TLR2, 4, and 5 from the animal that we start with. So to do a big sum up of all the constructs that we have, we need a lot of constructs to analyze what we want. And as you can see in gray, you can see that we can already find some commercially expressing vector, but at the starting of the study, we miss some constructs. So Nathalie Verling in the RVC construct all these expressing vector with the TLR2, TLR4, MD2, CD14, and the TLR5. So she made 18 new constructions, so it's a lot. It's a big work, and after that, I will test it in my functional test. So to test all these constructs, we use the X cells, blue KD TLR5. So it's the cell derived from the egg cells, so human embryonic kidney cells. And they are knocked down for the TLR5 to avoid to have any background in the test. And they are NF-kappa B inductive CYP reporter gene. So it said that when the cells are activating through the pathway of NF-kappa B, they can metabolize the medium, and the medium will turn to the pink to blue. So what was my test? So I see the cells. I transfected with the P101 construct that Nathalie sent to me. And after that, I collect my cells expressing the receptor, and I stimulate with Leptospira to see if they're activating the TLR through the NF-kappa B pathway. And if it's activating with the reporter system, we can see if the medium turn blue. It said that it's activating. So to show you some results for the TLR2, so you can find the TLR2 in two different heterodimers, so with the TLR2-6 and the TLR2-1. And in the example of Leptospira, the lipoprotein are recognized by the TLR2, TLR1. So we have all the construct of all the animals that we want to study, and I will show you just two results because it's too much if I show you all the graph. But here you can see that it's the play that I can obtain in my test. So again, if it's pink, it's not activated. If it's blue, it's activating. And what we can see here for the horse and the hamster, first we can see that the PAM2 and the PAM3, so it's the synthetic ligand positive control for the activation of the TLR2-6 and the TLR2-1 respectively. So we can see that the construct is working. And after that, we want to see with different tests of the four strain of leptospirosis that I present you just before. So Manille, Fiocruz, Verdun, and Pathoc. And for example, here in the horse, we can see that Manille, Fiocruz, and Pathoc, and I sum up this result in the tab here. We can see that all these three strains can be recognized at the same level. But we can see that, for example, with the Verdun strain, we have a low recognition or less than the other strain. In the hamster, it's the same, not the same, but it's almost the same. We can see that Manille, I will start with Pathoc one. So Pathoc strain, you can see that it's the most recognized strain in the hamster. And after that, we can see that with Manille and Fiocruz, we have the same level of recognition. And again, we can see here that with the Verdun strain, we have less recognized strain. And as you see here, it's representative summary of 3 experiments. And one time it's like very less recognized, and one time it's not. So with Verdun, we have very less recognition. So to sum up, we do all the tests with all the animals. So what we can see with this tab, it's when we conclude to have an overview of this TLR2 recognition with the live bacteria, we can say that the TLR2 show the most diverse recognition across the species. So in each species, we can see that different level of recognition depending the strain. And Verdun strain, so in this column, you can see that it appears to be the least recognized or not recognized at all of all the species. So this was a good result for us because we want to study more why this strain is like this. To conclude for another result, for example, you can see that the Pathoc strain, so it's the non-pathogenic strain that we use, we can see that this strain is very well recognized in all the species. So to go further with this TLR2 study receptor, we need to test the rat construction. So I already obtained it, but I know that it's working because with my positive control, it's working, but I need to plan the test with leptospirosis now. And to go further also, we want to test different MOI of leptospirosis, because for now, I use 100 MOI and we know that it's a lot for the cells. So we want to know if we decrease the MOI, we can see difference or modulation of this reconnection. So to move on to the TLR4 result now. So just to let you know, but you already see it in the first tab, for the TLR4 receptor, we need a trio. So we need MD2 and CD14 also. So to have the good construction, we need the three of them. And again, we have all the construct with all the animals, and the TLR4 recognized the LPS, as Catherine already mentioned, the LPS of the Leptospira. So I will not present you the result in the graph, but you already see it just before. But when we put the table of the overview of the TLR4 result, we can see that, for example, the bovine and the hamster here respond differently to LPS of lepto, with the modulation depending of the strain that we use. And the porcine, the mice, the rat here, and the equine recognize all the Leptospira LPS equally. And for the human, the TLR4 doesn't recognize LPS, and as it is known, and as it was published by the team of Catherine. So to go further in this TLR4 receptor, we want to test again a different concentration of Leptospira LPS to know if we can have some modulation or no in this recognition of the TLR4 for the LPS. So move on to the TLR5 results. Again, we have the same tab, we have all the construct, and the TLR5 can recognize the flagellin, but as Catherine said, Leptospira has the endoflagella. So when the bacteria is live, and it was published in this paper of the Catherine team, so we saw that when the bacteria is live and when it's sensing the TLR5, it cannot recognize because the endoflagella it's not outer the membrane. But when it killed the bacteria, and when we released this flagellin and this endoflagella, they can sense the TLR5 receptor. So all the results that I will show you for the TLR5, it's when the bacteria is killed and not live. So to sum up with the TLR5 result, we can see that so I already said, all the recognition it's with the kill Leptospira. And what we can see, it's for the bovine, porcine, hamster, and human TLR5, recognize all the Leptospira equally. And for example, for the equine, we can see that the TLR5 respond differently to leptospirosis, and we have some modulation inside the TLR5 equine depending of the strain that we use. And for the mice TLR5, they respond only to Fiocruz, so it's Leptospira interrogans Fiocruz, and it was consistent with what Catherine already published in the lab. To go further with this TLR5 receptor, I already obtained the rat TLR5 now. So I know that it's working with my positive control, but again, like the TLR2, I need to test it with the Leptospira reconnection. So that it's all the table that I can sum up for you, and it's a lot, but we can have an overview of the recognition of Leptospira and confirm that the recognition can be different following strain or host. And this result will enable us to establish correlation between Leptospirosa strain and the various species that we studied. And for example, the Verdun ictero strain is associated with severe disease in human, yet has no effect in rat due to their resistance. So we can study different hosts and different strains to see if we have modulation of recognition and the disease associated. We also hope that this approach and the new TLR expression vector that we obtain, which may become available, could be used by other scientists to study the interaction of PRR in different zoonoses. So we would like to thank our partner, of course, and the European Unions and the financial agency of the different countries that support this project. And of course, if you have any question with Catherine, we will be happy to answer them.
Megan, thank you very much for such an interesting and insightful presentation. Catherine and Megan, thank you both very, very much. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Dr. Wirtz or Dr. Fernandez at any point. Just while we're waiting, Catherine, Megan, for questions to potential questions to come in from our audience, I was just wondering, I mean, this is just a very new topic to me, and I imagine it will be very new to some members of our audience joining us this morning. And I was just wondering, in light of that, if we could maybe just roll back a little bit and kind of explore the kind of fundamentals a little bit, just for a minute or two. And I'm very curious, particularly as to why Leptospira interrogans is such an important bacteria to study in the context of animal to human diseases. I was wondering if you could maybe just explain that a little bit more for us.
So if I understand your question, is why Leptospira is so first, it's a worldwide disease. We can find Leptospira everywhere in the globe. But it likes rain. It likes to be in the soil, in the environment. So it's really the incidence of the disease for human, at least, is really important in the tropical area. But we still have in our countries lots of Leptospira. Most of the infections remains silent. So if you're not interested in it, for many years, people were having animals with morbidity, abortions, but they didn't know that it was leptospirosis. Now we are more aware of that, and we know that in the cattle and the breeding, it's a problem. It's a problem where the animals are not in good health, because most of the time, and for all animals, I think they can defend themselves properly with this immune system. Like the cells, the macrophage, are armed to fight leptospirosis, more or less. Then when you have these toll-like receptors activated, what we forgot to say is that it will leads to the cytokine production. The cytokines will lead to alert other cells, and you will have all the systems we will come to fight this bacteria. So why Leptospira is escaping the immune system, which is really well organized for all the disease, but it can go through. This is a question that I've been working for 30 years. And what I can see is that it's really, really well organized, and the strategy of we can't say a strategy of the bug, but it has been evolving with all animals and humans for for a very long time. It was there before the mammals happens. We know now that the bug is able to infect everything. So it has evolved with us, and we know that we may carry, even people in good health may carry Leptospira in their kidneys without knowing it. So for all the animals, it's an important thing to know, because then you don't understand why your animals are not in good health. And voilà. So because it's a neglected disease, it's not even cited as such by the WHO. The WHO, I don't know, World Health Organization. And we are fighting to make it a neglected disease, because it's over-neglected. And it's neglected because as there is 300 serovars, the literature is not homogeneous about this. It's not only Leptospira interrogans. There is so many serovars that the literature is splitted. So I think it's really important to publicize this disease. In France, it's still a disease that kills 10% of the people coming at the hospital because no one knows. Even the MDs are not aware. In our countries, it's a disease of the in people in slaughterhouses or in contact with animals. But it's not a very, the incidence is really low. So in France or Ireland, I don't know that, well, there are not so many people affected. So it goes under the radar.
Catherine, that's absolutely fascinating. And I suppose, just to follow on from that, bearing in mind Megan's kind of really interesting presentation about the different strains of the bacteria. And I was just kind of wondering about what role do differences in immune systems themselves play in deciding which animals get sick and which animals just, for example, carry the disease.
That's the purpose of our grant. And the purpose of one's life of research. We will be very pleased to be able to make these links. It's not obvious. Yeah. I am sure, though I already showed that it escapes the intracellular receptors, it escapes the pyroptosis, that usually when bacteria enter macrophages or stimulate the inflammasome, they are killing the cells, and then the cells release a lot of things that are augmenting the immune response. But Leptospira doesn't do that. Leptospira keep the cells intact, and then it avoids big inflammation. So this bacteria is really an anti-inflammatory, and we showed it already, is really anti-inflammatory. And then your immune system is not able to fight because everything is under, it's not inflammatory. But I don't think it's one mechanism. It's an accumulation of diverse mechanisms. That's why we wanted to do this catalog and understand better for the different animals, because the equine with human are the only one to have this uveitis. Why? Then we have to get interested in the toll-like receptors and not-like receptors expressed in those specific organs: eye, kidney, liver, lungs.
It's fascinating stuff, Catherine. Thank you so much. And I was just lastly, I suppose, just before we move on to Mariette's talk, I was just wondering about, plans for the future, looking ahead. What are the kind of next big questions that your team plans to look at? Do you plan to, for example, test these findings in whole animals and not just in cells, or what are the kind of future directions, if you like, of this project?
So we are really interested in the bovine because we have already murine models. They are really close to what we can have in human disease, but we don't know a lot about bovine thing. And then with INRAE in France, we can maybe goes more into this host. So first, studying what's happening in blood, like we are doing in human patients as well. But yes, and then our question is how to overcome this downregulation of the immune system, like if we can push back the immune system to defend itself. So it's all these host-directed strategies that we started in the lab that we are following now.
Excellent, fascinating stuff. Catherine, Megan, just lastly, one or two questions coming in from our audience. Sabine asks, is it known if the genes associated with these evasive mechanisms belong to lepto, or are they part of the genome that gets shared with other bacteria?
So there is very few virulent genes identified. They are part of the LPS biosynthetic pathway. They are part of, there is methylase. There is a lot of different things very specific to Leptospira. There is still a lot of genes unique to Leptospira that have no autologs. So, and there is some genes that maybe, but most of the genes are specific to Leptospira. And as genetic tools are really, we can't just mutate what we want in Leptospira. It's still very, very difficult to do reverse genetics. So the study is really behind compared to the other bacteria. The horizontal transfers of genes are really, really important in Leptospira. And they can share genes with most probably other bacteria, but it's not known. Thank you very much.
Catherine, Megan, thank you so much again for your really excellent presentation, and thank you to the audience for your questions. I think we could go on and go on with more questions for you both, but unfortunately, because of time, we'll have to leave it there. Okay, so now joining us is Dr. Mariette Ducatez, a senior virologist with over 15 years of experience studying animal and zoonotic respiratory viruses at INRAE in France. Having trained as an agricultural engineer, earned her PhD, and completed research positions in Luxembourg and the United States before joining INRAE's IHAP unit in 2012. Dr. Ducatez's work focuses on influenza viruses at the human-animal interface, as well as other emerging pathogens affecting livestock. At INRAE, she leads programs on viral ecology, evolution, and transmission, including extensive surveillance networks in Africa and Europe. Dr. Ducatez has coordinated numerous international projects on zoonotic risk assessment, pathogenesis, and prevention, and is recognized for her experience in influenza C and D viruses. Today, Dr. Ducatez's talk is entitled Longitudinal Study of Influenza D Virus and Co-infecting Pathogens in French and Italian veal calf farms, which is also part of the EU-funded ICRAD initiative. Mariette, thank you very much for accepting our invitation to our webinar series. Again, like Catherine and Megan, we're really delighted to have you joining us today. So without further ado, the floor is yours. I'm going to stop sharing my screen and you can start sharing yours. Thank you very much.
Thanks a lot, Barry, for both the invitation and the introduction. Very nice. Right. So yeah, thanks a lot. As you mentioned, this the project I'm going to present today is indeed part of an ICRAD-funded project as well. So the partners here are, so I'm based in France at INRAE in a joint research unit with the vet school in Toulouse. And we had partners from 2 Swedish labs at SLU and SVA, from the University of Liège in Belgium, the University of Istanbul in Turkey, as well as from the Istituto Zooprofilactico of Parma and Brescia in Italy. And for the sake of time, we've selected as well just a tiny part of the project to present, and the data I'm going to show you are more from France and Italy. So I'm going to start with a big thank you to Anna Moreno from IZS in Brescia. So our ICRAD project is entitled Preventer, for deciphering the role of influenza D virus in bovine and human respiratory diseases in Europe. And we had 3 work packages, one being more surveillance, one being more mechanistic, trying to see in vitro, ex vivo, and in vivo how do pathogens interact either in synergy or in antagonism somehow. And our 3rd work package was more epidemiology, trying to predict the incursion of influenza D virus in human. So I'm just going to touch on the very beginning, actually, of our project that finished early 2025, which is the active surveillance we carried out in cattle farms, in veal calf farms, to be more precise, with respiratory diseases. And we worked in France, Italy, and Turkey, but I'll focus on France and Italy. And before moving to the data itself, I'm not sure how well known influenza D virus is, but it's been discovered just a bit less than 15 years ago, first in the US. That's the first paper describing it. And it was identified thanks to influenza-like illness in pigs at the beginning in Oklahoma in the US. And all the respiratory pathogens had been tested, but none of them could be identified. So it's really electronic microscopy studies as well as next-gen sequencing that allowed us to identify this new orthomyxovirus. So you have the first EM pictures down there from the first paper, and next-gen sequencing allowed to identify 7 RNA segments that were orthomyxovirus-like in terms of genome similarity, identity, it was just about 50% overall homology with influenza C viruses of human origin. And the first serological analysis that were carried out at the time showed that antibodies generated against this new virus did not cross-react with influenza A, B, or C, which is what really led to the new genus influenza D virus. In terms of circulation in the world and host spectrum, the virus or its antibodies have actually been detected virtually anywhere they've been looked for. So on this map that's actually not fully updated, you have the detection of the virus or genetic material nucleic acids in blue, in yellow detection of antibodies alone, and in green it's when both antibodies and virus have been detected and identified, characterized in most cases. So you see Asia, Europe, the Americas in general, we have the virus in Africa as well. So that's really very wide geographical tropism. And what we know from the last 15 years of work on this is that cattle seems to be the main host. We are not saying reservoir yet because we've not investigated it all at all. But then virus or antibodies, specific antibodies have also been detected in equine, camelids, goat, sheep, swine, and human. And looking a bit more into the genetic characterization, so a bit of phylogeny of the virus, just looking at the surface glycoprotein gene, which is the hemagglutinin esterase fusion gene, We have two main genotypes, the D. oklahoma, like the original strain gave the name to this genotype in red, and in blue, the D660 like lineage that do circulate both in the US, in North America, and in Europe. And then we have smaller genotypes, for instance, down here, two of them only circulate in Japan as far as we know so far. And within the framework of our European consortium, we've been able to date a few years ago the emergence of both D-OC and D660 genotypes in Europe. So we think the most common research, the most common recent ancestor to the D-OC genotype was early 2013 in Europe, and a year and a half later for the D660 genotype. But from there, so we knew we had a description somehow, something quite descriptive for the virus, but we did not know if IDV would lead to disease, clinical signs per se. We did not know how long and in which tissues it would replicate, how it would transmit, and how the immune response would be. So that was a really nice project also for young scientists to start with, because there was a lot of unknowns as far as the big questions are concerned somehow. So I'm going to very quickly show you data from an experimental infection we carried out a few years ago with 2 to 6 weeks old calves that were colostrum deprived to avoid having maternally derived antibodies because the seroprevalence is really high. And we had a group here on the right-hand side inoculated with a high dose of virus, 10 to the 7 TCID50 per calf. And 3 meters away, we had an aerosol contact calves group that could not touch at all, of course, their counterpart here, with the air flux going from the inoculated to the contact, and barriers and personnel that was different on the two sides. And in a separate building, we had the sentinel calves non-inoculated, not in contact at all. So we looked first at respiratory clinical signs. I'm going to go really quickly to be able to show you a bit of the ICRAD results, but we saw mild clinical signs in the inoculated group only, virtually, with a peak around a week. Then we looked at the shedding, so first in the upper respiratory tract, looking at nasal swab samples, and we saw really good replication that lasted at least 10 days, 2 weeks for some animals here, with a peak around 5 days. And we also could detect virus in the lower respiratory tract thanks to bronchoalveolar lavages that we did at day 2, 7, and 14. So the virus is still in the lungs 2 weeks post-infection. And then we looked at the transmission. As I mentioned, the setup was made for it. And here we have the same plot with time on the x-axis and RNA copies. Sorry, I did not walk you through very well before. In blue, you have the inoculated group as before. And that's the mean of the 8 animals. And in the 3 shades of greenish, we have each time 1 aerosol contact animal. So what we've observed is that we have had transmission, at least to this bright green first contact animal. We believe it's aerosol transmission because that's also supported by the Coriolis air samples that we took every other day in the area, so either in the inoculated area, in the buffer separating lane, or in the contact area. And in the contact area here, for instance, we could detect virus in the air 5 days post-challenge, post-infection. So we believe that having virus from day 11 detected in 1 contact animal is really the result of the infection a few days before. However, we think the other 2 animals were actually in contact directly with IDV from the bright green individual. So to conclude the state of the art spot somehow, right now we know that IDV is present worldwide with a very large host spectrum. It's been associated with some clinical signs in the field, but we've also showed in experimental conditions that it does induce mild disease, but disease that a farmer would not necessarily really notice that much if IDV were the only pathogen present. It does replicate both in the upper and lower respiratory tracts in calves and transmits both by direct and aerosol routes. And I did not mention the seroconversion, but by 10 days post-infection, we did see antibodies. So when we started this ICRAD project, we really had 4 questions. First of all, which is the role for IDV in respiratory disease, and how does it interact with co-infecting respiratory pathogens? Because in cattle, it's really well known that usually you have a cocktail of pathogens. It's called the bovine respiratory complex disease, which by definition is complex, and it can be caused by a handful of different pathogens. We also wondered how long the anti-IDV antibodies would last. So for this, as I mentioned initially, we had 2 cohorts, and I'm going to even show you data just for 1 farm per country in the interest of time. So first, the French cohort, we had veal calf farms, and we tried to homogenize our sampling and screening strategies in the 3 countries where this study was carried out. So we had animals younger than 1 year of age. We took nasal swabs and blood samples for the interest of the data I'm going to show you here, with 8 sampling times. At the beginning, it was day 0, 1 week, 2 weeks later, and then a month, all the way to 5 months virtually, for 30 animals. And we screened nasal swabs by RT-qPCRs for 7-8 classical respiratory pathogens, and did hemagglutination inhibition assays to look at serum antibodies. And here, sorry, the slide is really busy, but what I want to show you is a bit of a summary of the nasal swabs RT-PCR screening results we got, each line being 1 calf that we followed then throughout the 5 months. And then you have each box is 1 time point. So you have day 0 first, day 7, 15, 29, and this is 5 months. And the redder the color, the more positive, so the higher the viral load in terms of RNA copies or DNA copies. So if we look at IDV, which was our focus for the project, we just had 1 very weakly positive animal initially, then 2, but it's really at the month of age that the 2/3 of the animals were positive, and then the infection was cleared. So we did experience really an outbreak of IDV during our longitudinal study. And we also looked at other pathogens. What I want to highlight is that the bovine coronavirus B curve here is extremely prevalent, with all the animals were positive initially, and then it stayed quite a bit before clearance but it was never completely cleared. It's as if some animals were reinfected later on. And the pathogen that's just the column after that, Pasteurella multocida bacteria, is also extremely prevalent. It's a bit harder to tell because we know there is a bit of background noise in the assay. So high CT values by PCR can actually mean just chronic carriage. Somehow it does not mean it's associated with clinical signs, but at least we have a very high prevalence here. And that's a table summarizing at each time point the percentage of animals that were positive for one or the other of the pathogens. But I think I gave you the message already. And in terms of seroprevalence, there again, most of our animals were seropositive for IDV throughout the study period. Virtually, we only have a few animals that stayed seronegative for a short time, but they all ended up being positive at some stage. So one of our questions was to know how long would these anti-IDV antibodies last. And of course, as we only worked for 5 months, the maximum we could know is up to 5 months somehow. And as some of the animals were really only infected at a month of age, it's hard to be very precise, but we think at least the duration can be of a few months. And if we look in parallel at the study that was carried out in Italy, I'm going to go much faster because the study design is virtually the same. We did not have exactly the same types of tests, but in general, it's really extremely similar, same time points. And here we have in blue the virus prevalence for IDV. So there again, in that example for that farm, we did have an outbreak of IDV with half of the animals being infected at 2 weeks, a bit more than half at 4 weeks post the beginning of the follow-up and clearance of the virus. And in orange, you have the antibodies that were already higher at the beginning, of course, maternally derived antibodies, and even higher. We reached almost 80% at some stage. And now that's a bit of a different graphical visualization of the data, but we have all the respiratory pathogens that we know. And here again, you have the bovine coronavirus in blue that was extremely prevalent, close to 90% initially, and got resolved somehow. Mycoplasma bovis in green was the number 2 in that specific form, but Pasteurella multocida, which is the dark blue, was also present throughout the study period in the farm. So the main pathogens that are co-infecting seem to be quite similar. That's something we've seen in Turkey as well. And in terms of prevalence, persistence, of anti-IDV antibodies, here we know that 3/4 of the animals kept antibodies more than 60 days. The calculations are a bit different because not all the animals were seropositive at the same time, but we have that 2 months window at least. But initially, we see that animals that maybe it's even clearer, here, that some animals lose antibodies as well against IDV. So it does not look like once you've been infected once, you keep those antibodies for life. And what's also not known is whether they're going to protect the animals later on. So to conclude, we know that IDV widely circulates in France and in Italy, and almost always in co-infection. It's very rare that we find IDV alone, actually. The most common respiratory pathogens that have been detected in our study cohorts here were the bovine coronavirus, Mycoplasma bovis, and Pasteurella multocida. And we know that in veal forms, the duration of antibodies against IDV should be at least 2 months to maybe 5. But of course, the study was pretty short, so it's just an estimate. That's an at least as a number. So what we still need to do with this data is to run biostatistics to assess pathogens associations, see if there is a higher risk or a higher protective indicator. If 2 pathogens are found together, it's less likely maybe have a third one. So that's the perspective of this part of the study. And with this, I'd like to thank, of course, our research unit in Toulouse and everyone who's worked on the project, especially our ICRAD consortium with Chiara Ciapponi and Anna Moreno from Isla in Italy, from which some of the data has been presented here, and ICRAD for funding. And you for your attention, of course.
Mariette, excellent on the timing. Thank you very much for such an interesting and insightful presentation. I just want to remind everyone that the Q&A and chat box is open, so please do feel free if you have a question for Dr. Ducates at any point. Please do feel free to type it in. Mariette, I think you probably touched on this a little earlier in your presentation, but I was just really wondering about the kind of prevalence of influenza D in older cattle. I think you did say that it was there, but not as prevalent. So I was just wondering about how common is influenza D in older cattle, and should we be concerned about it spreading more widely?
It's actually what we know so far is if you do serology studies, depending on the country, you have 40 to 80% of the cattle older than 2 years of age that are seropositive. So it looks like most of them have seen the virus in their life, if you want. And we saw a difference between countries that tend to export cattle, like France, for instance, has a lower seroprevalence than countries importing cattle, like Italy or as an example, which had 80% seroprevalence in our case. But then if you look at the viral prevalence, so your probability of finding the virus, it's much higher in veal calves than in dairy cattle or just older cattle in general. So it looks like that's really when the animals are young that it circulates most.
Fascinating. And Mariette, is there any evidence that influenza D can spread to humans, or is it mainly an animal virus?
No, there is evidence. It's actually still hard to say for sure because there are pieces of evidence of antibodies against influenza D virus, but then no one has ever detected replicating infectious virus in human. The only studies found it in the air, or well, there are hints that suggest it may be there, but as long as we don't have an isolate, I will not say we are sure, but we do have the good receptors. In histology, you can do some staining and you can show that in theory the virus could come in. That should work, but we've never really observed a case. So we think it's indeed a potentially zoonotic virus. We don't believe that even if it went into humans, it would be a big problem either. But we still lack the virus itself in human beings.
Excellent. And just last question from me, Mariette. Were there any clear differences between French and Italian farms in terms of infection rates, or was it kind of very much similar in both regions?
In terms of infection rates for IDV, it's hard to tell because we just had 30 animals in both cases, and I've shown you the two farms with the nicest outbreak that arrived in the middle of our follow-up. Of course, in the other farms, in some cases, we did not have any IDV outbreak, and in some other cases, it's really the timing is less obvious, I would say. So maybe, no, I really don't think we can conclude on the incursion rate on the.
Yeah. Sure, would necessitate a much broader, wider study done over a long time.
Yeah,
I'm sure. Just one question coming in from the audience, from Sabine. I think I might have missed it. Were there thoracic lung scans done alongside clinical assessments? So she just wanted to know, Mariette, I just wanted to know if they ultrasounded the lungs alongside observing for clinical signs of disease.
No, for the study I've presented here, whether it was actually in the state-of-the-art part where we did the experimental infections or for the longitudinal studies, we did not do any interesting lung scans nor ultrasounds at all. In a more recent study for another project, we did include this in our experimental infection. So it seems to go along with the clinical signs. That's what I can tell you, but for the data I've presented here, no. not there.
Mariette, it's just gone past 10 o'clock. I'd like to thank you again for your impeccable timing, bringing things to a conclusion on time. So we really appreciate that very much. I'd just like to thank once again our three excellent panelists this morning, Dr. Catherine Mertz, Dr. Megan Fernandez, and Dr. Mariette Ducates, for giving us such wonderful and insightful presentations and providing us, I think, with tremendous overviews of their respective projects this morning. The next Breakfast Club webinar will be in 2 weeks' time on Thursday, September the 25th, and I'll be sending out the invitation to all of you in the coming days. Again, thank you very much for our panelists, and thanks everyone for attending and for your questions this morning. We really hope you enjoyed the webinar, and we really hope that you join us in 2 weeks' time for our next session. Thank you very much, everybody, and have a good morning.